This is our protein synthesis presentation. Proteins are the fundamental components of life. In structure, they are basically long polypeptide chains of amino acids formed in the ribosomes. The DNA contains the information needed to synthesize protein. However, the DNA cannot leave the nucleus. Instead, through transcription, a strand of messenger RNA, or mRNA, must be produced to carry the information.
Transcription.
Transcription begins similarly as replication. First, the enzyme helicase unzips the DNA into two strands. The code for each amino acid is contained in groups of 3 bases called triplet codes. The 5' to 3' strand in transcription is called the coding strand, or the sense strand. As its name suggests, the coding strand contains the information for protein synthesis. Reproducing this strand, however, requires the other strand, which runs in the 3' to 5' direction. This strand is called the template strand, or the antisense strand. Using the strand as the template, complementary base pairs are bound together by RNA polymerase, resulting in mRNA. There's only one small difference between the coding strand of DNA and the mRNA. In mRNA, thymine is replaced by uracil. Also, the codes for amino acids are still in groups of 3, but they are called codons instead of triplet codes in mRNA.
Before the mRNA can be transported outside the nucleus, it must first undergo 2 important steps. The first step is 5' capping and polyadenylation. A 5' cap is a methylated guanine nucleotide added to the 5' end of the mRNA. It signals the beginning of the mRNA and is critical for recognition by the ribosome, and also for the protection from enzyme degradation in the cytoplasm. Polyadenylation, on the other hand, is the addition of a chain of adenine nucleotides, called a poly A tail, at the 3' end of the mRNA. The poly A chain degrades in the cytoplasm in place of the mRNA. The second step is the removal of extraneous sections called introns, which do not code for protein. The remaining necessary sections, called exons, are spliced together by spliceosomes, forming mature mRNA. The second step of protein synthesis is amino acid activation. In amino acid activation, a different form of RNA plays a key part, the transfer RNA, or tRNA. tRNA molecules are shaped like clover leaves. At the base of each tRNA molecule is an anticodon, a 3-base sequence that complements a codon. Now, every codon codes for an amino acid. In the cytoplasm, when an amino acid locates the correct anticodon, it binds with that tRNA. This step is called activation, and it requires ATP and a specific enzyme depending on the amino acid. Although a tRNA molecule can only carry one specific amino acid, there may be up to 6 distinct tRNA molecules carrying the same amino acid. This is because of the degeneracy of the genetic code, meaning that some amino acids have several triplet codes.
Back to the mRNA. The strand of mRNA leaves the nucleus through a nuclear pore. Now, once amino acid activation is complete, the mRNA is ready for its final step,
Translation.
which takes place in the ribosomes. Ribosomes are made up of 2 subunits, which are composed of ribosomal RNA, or rRNA. mRNA is read as the ribosome moves along the mRNA in the 5' to 3' direction. The larger of the 2 subunits is the one where translation actually takes place. Once the ribosome reads a codon, the complementary tRNA enters it. This step of translation is called
Initiation.
There are 3 sites in the larger subunit of the ribosome: the A site, the P site, and the E site. The tRNA enters through the A site. In the P site, its amino acid is taken and polymerized to form a part in the polypeptide chain. This step is called
Elongation.
Finally, the used tRNA exits through the E site to be reused in the cytoplasm. The polypeptide chain formed in the P site, once completed, is the protein and is released into the cytoplasm. This final step is called
Termination.
termination.
This ends our protein synthesis presentation.
Thank you very much.